And I will send you guys a link to the recording once it's done. Yeah, so basically there's a little recording button, and when we're done, turn that off, or we'll shut down. Yeah, and then if somebody wouldn't mind just bringing our sign in and sticking it back in the corner when we're finished. Yeah, after we're done, just in case other people show up. Good, but I think that'll take care of you guys for the moment. That's good. I'm gonna just adjust the lights so that it's a little easier to see. And we do have members who check our videos and stuff, so even if we don't get that many more people here, we will have the video to share, so that'll be good. Okay, you think you're good?
Just a question about how to advance slides.
That's a good question, because I kind of brought you up in a different way. Let's see.
Also have the video, so oh yeah, there. Oh, so it's
yeah. So now it's looking. It was just not. I had not chosen that guy. Okay. Perfect. All right. Thank you both so much. Thank you for being here. Hopefully we'll have another person or two, and we'll have a video. So very good.
Should I get started, or okay, you can go. Sure, I can get started. So my name is Gapinski. I am a faculty member at Bellevue College right now, and I've been here for about 9 years. So today I was going to start off just by telling you a little bit about my journey and my career so far. And then I am the program chair for the Molecular Bioscience program. So I was going to tell you a little bit about that program as well. So advance. So I share with my students sort of a geographical history, I guess, how I got to where I am today. So I was born in, if you want to click, I have a few animations.
Okay.
I was born in Missoula, Montana. So I grew up in Montana. And I always was interested in math and science as a kid. I thought I wanted to be a doctor. So I started off my journey with the plan that I wanted to be pre-med. And I thought I wanted to be a pediatrician. I had it all figured out. And then for undergrad, I just looked at 2 different schools. At the time I applied, I wanted to get away from home, so I didn't apply to any school in Montana, but I wanted to stay somewhat close. So I applied to 2 schools in Washington, Gonzaga and, yeah, it's fine, this is fine. It kind of gets to the punchline, Gonzaga and UW. And I visited, and really what I liked about Gonzaga was how small it was, and that's kind of a theme that, as you'll see throughout my journey, I really like when in the educational setting, when the faculty get to know the students. And so that's kind of why I chose Gonzaga. But I went there, and again, with my plan was to be a doctor. So I was planning on majoring in biology, and I started doing some kind of volunteer work in the hospitals and observing. And I had a couple of times when I fainted in the hospital setting when I was observing for procedures. It wasn't really because of the procedure itself. I wasn't like squeamish with blood or anything, but I didn't like, it was always in a situation where the patient was in a lot of pain and they had to do a procedure and they couldn't really address the pain. And so, and all the smells and everything, and then I would just faint. And then I would wake up and I would be surrounded by all these healthcare providers that were trying to figure out why I passed out. So I started to think, maybe that's not the right career for me. I thought about nursing, but again, nursing and they're very much the same setting. So then I did some research over, I think my sophomore year summer, I did some research with a faculty member, and I started to think maybe I could be involved in medical field in a different way by trying to do research. So the next slide, one more click forward. Oh,
sorry, that was backwards.
Yeah, so what I decided, one of my faculty members had told me about this program through the National Science Foundation, this research experience for undergraduates. And so I looked on this website, and they had all of these different internship programs across the country. And I was really interested at that time. I thought I wanted to do cancer research because I thought that's still close to helping people, but maybe in a more indirect way. And so I applied to a program that was in Buffalo, New York, and it was at Roswell Park Cancer Institute. Very similar to Fred Hutch or MD Anderson, so it's just a large research institute that has many different labs studying different types of cancer. So that was a really formative experience for me because I actually got to see what it was like to do research in like a real lab. I wouldn't say that Gonzaga wasn't a real lab, but it's just a very different setting. And it was a 10-week internship. We stayed in the dorms. So we had this cohort of students and really got to see what it was like to do. You don't get a lot done in 10 weeks, but I was exposed to kind of the culture of research. And that kind of made me switch my direction and think maybe I should actually go to graduate school when I'm done with my undergrad degree. So flash back to, I was still finishing up at Gonzaga. I had thought about taking a year off. Gonzaga is really all about like the education of the whole person and providing service. So I had briefly considered maybe I should take a year and do like a volunteer tour. But at the end, I decided I have momentum and I don't want to take that time off. So I applied directly to grad school right after graduating or in that senior year so that I could go in the following year to grad school. And again, I applied to UW, a program at UW, and also to Johns Hopkins. And I actually applied to 2 programs in the School of Public Health at Johns Hopkins, and they were both, one of them was a basic science. It was biochemistry and molecular biology. The other one was microbiology and immunology department. But again, both of them were in the School of Public Health. So I still kind of had in my mind that I wanted to get the framework for like systemic change, I guess, and being able to make change at a systemic level. So you can click forward. Oh, that's the website for that. If anyone is interested in applying, I don't know what's going to happen to that program with the administration now that we have, but it was still up and running. And so it is a really good experience for undergrads. So anyway, there's the link for that. But then, yes, so this was the Bloomberg School of Public Health. And then on the next slide, so I spent 6 years living in Baltimore and going to school in this building, which is the School of Public Health building right across the street from the hospital. I lived in Fells Point, which if you've ever seen Sleepless in Seattle, that's where they are. And got to do all these things in Baltimore. For me, it was a really good experience kind of living on the East Coast. It's culturally very different than the West Coast. But, grad school for me, I look back on it, it was a really good time. It was also hard, but I feel like I made a lot of lifetime friends, and it was kind of like you kind of struggled together. So it brings you together. And the faculty that I got to know during that time, they were phenomenal people, just very passionate about their research. And so that's sort of my grad school years. I will say a little bit about what I studied during that time. I joined a lab that was studying a protein that's called SUMO, which stands for small ubiquitin related modifier. Some people have heard of ubiquitin because ubiquitin is a protein involved in degradation. That's probably its most famous role. But SUMO is sort of the little cousin of ubiquitin, and they were still figuring out, and they still are figuring out what exactly SUMO does. So it gets covalently linked to other proteins, and then it serves as like a mark for something, and it is sort of context dependent. But that's what my lab did. During grad school, I learned some new skills. Mostly we were working with mammalian cells, so we were doing tissue culture and developing reagents for use in tissue culture. I didn't really have experience of doing any like mouse work or anything during that time. But my goal, as I was in grad school, I started to realize that I really liked teaching. So I had to do some teaching assistantships during that time. And I kind of decided, after I get my PhD, I do want to eventually find a professor position. And at the time, I thought I wanted to go back to a place similar to Gonzaga, just so there's lots of small liberal arts universities. And I was thinking that would be where I wanted to work. At that time, I had very little exposure to community college. So I will kind of tell you how I shifted and ended up at a community college and ended up loving this environment. But that was my plan. So after grad school, I thought I needed to learn a new system, a model organism or something that would be amenable to undergraduate research. So in my postdoc, many people will do a postdoc after they graduate. And I went from Hopkins down the road to Bethesda, Maryland to the NIH. So the NIH has not only provides funding for all of the different labs across the United States, but also has many labs in Bethesda. Those are intramural labs doing lots of different research. And so I found a lab. I actually only looked at labs where they used some kind of a model organism. And I was really interested in C. elegans. I don't know if you all heard of C. elegans, but they are small worms. They are nematodes. And they are used as a genetic model organism. So I ended up in a lab that studies C. elegans. I think on the next slide, yes, you can play the video. Click on that. So this little tiny worm, it's actually 1 millimeter in length. So if you, by naked eye, you can barely see it, looks like a comma. But the reason why it is really useful to study, you can see that if you click pause, you can see that it's transparent. So it's easy to actually visualize structures within the worm. It's also self-fertilizing. So it's a hermaphrodite. It's very interesting. It starts out as a male and it produces all the sperm that it will ever use. And it stores them in a little compartment called the spermatheca. And then it switches its developmental program to start creating oocytes. And if you go to the next slide, I can show you just a still shot. Oh, I has to go all the way through. Okay, yeah, so this is a still shot, but you can see it has two U-shaped gonads, and those at this point in time, this is an adult, so now this is producing oocytes. So there's sort of a mitotic zone and then a meiotic zone. This is where the spermatheca is, so all those sperm have been stored up. And then basically these oocytes pass through, get fertilized, and they start growing and dividing in the uterus. And then they are laid out at about the 30 cell stage and they continue developing until they hatch. This egg is actually a really useful system for studying early embryonic divisions. And so in the lab that I joined, we were actually looking at that embryo stage and looking at a structure inside called the centrosome. So if you take in cell bio, you might remember centrosomes organize DNA. If you go to the next slide, this is a single, so this is basically the outline of a single embryo that has been just fertilized. And one of the reasons why C. elegans is very useful as a model organism is that we can manipulate all of its genes. We can look at fluorescently labeled. We can actually add these fluorescent proteins to look at the location of different proteins. And with this example, you can see in real time what is happening with centrosome duplication. So in the lab that I joined, we studied centrosomes and what regulates their duplication. So if you push play on this video, this is a wild type. Oh no, maybe the videos didn't come. That's okay. I guess they didn't get saved when I said that. But what happens is you see these two green spots, those are the centrosome markers. And if you watch, we essentially took time-lapse microscopy, used time-lapse microscopy to take many images, put them together in a video. And you can see that both of these divide into two. So as this continues to develop, you will always see that centrosomes get duplicated just like DNA every single cycle, so that every cell division event has two centrosomes to divide the DNA. In this next one, which unfortunately you don't get to see what happens, we have a mutant, the Zy1 mutant that is missing a critical factor for that cell division. And so since that can't happen, these embryos get stuck in this very early stage. They can't divide because they have only one centrosome, so there's nothing to pull the chromosomes apart. So this mutation is actually lethal. And it was first discovered in worms, but then it was found that there are orthologs in humans. And so part of the reason my lab was interested in this and how we got funding from the federal government to study this process in worms is that many cancers actually have problems with their centrosomes. So the next slide shows a table from a pretty old article, but showing that all of these different cancer types, they either have numerical problems with their centrosomes or they have structural problems. And for many cases, it is found in early lesions. And not to say that this is causative, that that's causing, but it definitely is something that is observed in human cancer. So this is one of the things, one of the reasons why we can justify studying centrosome duplication is to further be able to understand what's going on in cancers when there is sort of misregulation of centrosomes. So that's what I sort of learned about during my postdoc. And during that time, I also taught classes kind of on the side. And that's really when I was sort of formalizing that I enjoy the teaching aspect. I like research, but I really liked working with students with research, and also whenever I was teaching, I kind of loved spending the time prepping for my classes. So at this point in time, I decided that my next position, I wanted to focus on teaching. And so I then moved back to the West Coast, I think on my next slide. And that's when I didn't know a lot about community colleges at all. And I started out just thinking I'll get my feet wet just teaching. And I taught whatever they needed me to teach. I actually started out teaching nutrition. But once I came to Bellevue College and sort of began, I really enjoyed the students. I really loved how diverse the population was and how really the college was so focused on helping students kind of find their path and meet their goals. And so ever since, I've been a huge proponent of community college, and this is really sort of my dream job. And then Bellevue started a bachelor's program when I was here, and so I helped work on developing some of the classes for that. And I think on the next slide, yeah, so I was going to talk about some different possible careers and different careers that I've seen, you know, some of my colleagues go into. I think getting a degree in biology, molecular biology, or biochemistry can open a lot of different types of doors. And many of these jobs would require a PhD, but not all. So I've definitely seen people get into these positions with a bachelor's as well. But there's kind of different routes. If a person really likes research, there are many jobs in research labs that are sort of in the academia path, which is, you know, looking for jobs in universities, and they hire research techs, they hire lab managers, lots of different positions like that. I personally never worked in the industry, but that is definitely a route that many of our current MBS students, they find jobs in biotech companies. And so, or you could go into pharmaceutical companies, biotech companies, so lots of different possibilities there. The one thing I've heard about the difference between those two routes, either academia or industry, is kind of what drives the research. So I think in academics, there's some freedom there as long as the PI is able to get funding. They do their main research that they're funded for, but I think a lot of them have passions and they might have a side project they're working on at the same time. As long as they can fund their lab, they have a little bit of academic freedom. Whereas what I've heard about industry is that it is driven by, you know, what is going to be most successful. And, you know, if it's ever the case that this therapy is hitting some roadblocks, I think sometimes decisions are made to drop that project and follow the most promising lead. So, you know, people who I've known to have worked in industry sometimes have had projects that they've said, okay, we're going to shift gears now and work on this. And then you have to shift gears. So that would be the only, you know, that's a difference that I can see. Our students that are, you know, in the industry route, I think they get paid pretty well. I mean, our students have a bachelor's and then they're getting lab tech positions, and some of our students have been promoted to sort of higher, you know, they've kind of climbed and been promoted to oversee a section of a lab or things like that. So I think there's a lot of potential for growth in that role in industry. With academics, I think you're limited if you want to be a principal investigator, you almost have to. You have to go the route of PhD, a postdoc, secure your own lab, and so it is a little bit more rigid in how you climb the ladder. But there's a lot of other jobs outside of just lab jobs, because sometimes people say, oh, I realized I didn't really like lab work that much. So some of my friends or colleagues from grad school went into policy. And part of this was because where I went to school was very close to DC. So we heard about fellowships that were available. But some of them got involved in nonprofit organizations. Or working for like think tanks and helping to advise or advocate for certain science policies. I have several people that I know that went sort of in a science writing route. They were very good writers and didn't necessarily enjoy lab work. And that there's a lot of different possibilities. You can either be a freelance, do this freelance article by article, or some of them will be hired as editors for journals, and they will be writing pieces that are sort of the preface to different journal articles. Or even there's positions at places like Fred Hutch for writing these press releases that go out to the public about the research that's being done. So that's kind of an interesting career offshoot if you end up not really enjoying the actual lab research. Some people have gone into the outreach branch if they enjoy education, and especially if they like informal education. Many cities have a science museum. We have one here. There's also after school programs. And I actually worked for an engineering for kids after school program when I first moved out here, partially because I just had a baby and I was like, I just want something very short and then get my feet wet again. So there's lots of possibilities with that. And then the last two I have here, I've known some people that enjoy or wanted to get into patents, and there's sort of this crossover between science knowledge and then law. We've actually had a student in MBS who had a law degree, was a practicing lawyer, and what he wanted to get was the science background to be hired on as a patent attorney for a company. But I have seen people, one person from my cohort in grad school, he ended up stopping the PhD program at the master's level, and then he went to law school because he wanted to practice patent law. So there is that crossover, and you don't need a law degree. You can also apply to work for the US Patent Office as a scientist, and they train you sort of on how to review patents. So that's a possibility. Government, there are these federal agencies that are located in different places. Right now I have a person from my lab is working for the CDC. I'm really curious how things are going right now. It's been like a very prolific thing. She worked on meningitis for a while in that section. And then during COVID, she actually was working for the branch that was giving messages out from the CDC. And now I'm not exactly sure where she's been assigned or what's been going on with that, but she got that initial position by doing a fellowship at CDC, and then actually got her foot in the door to apply for a position. I also have another person from my graduate school lab who's working for the FDA right now, and her section is the section that looks at genetically modified organisms. So kind of looking at whether they should be approved, get FDA approval. And then, I have many people just having worked at NIH that still work in labs at NIH. And there are, I think, something like 30 institutes. So there's like National Cancer Institute. I worked for the NIDDK, which is a diabetes and digestive kidney disease section, but there's lots of different institutes there still. And then if you're kind of interested in environmental science, there's EPA. So lots of possibilities depending on what your interests are. On my next slide, I have kind of pros and cons, or education and qualifications, and then pros and cons. I would say that if you're not sure exactly what you ultimately want to do, starting with the bachelor's degree, for most technician jobs, although we offer a 2-year associate's degree, I think in most places it helps to have a bachelor's to be competitive. And so, like I said, for most of our students, they work as lab techs, or their titles are research associate or lab tech or research tech. So I think that's a good starting point. If you get in and you realize, okay, in order to really move up, I need a master's or a PhD. And in some cases, postdoctoral training, like if you want to have a lab yourself, a lot of times it's gonna be a lot of years. It's, the bachelor's, PhD, I was at Hopkins for 6 years. That was kind of average in our department for how long it took people to finish. And then a postdoc, I think they try to limit them, but some people will spend 5 years in a postdoc before they get into, if they want to have their own lab. So it is a long road, but I will say one kind of positive is that if you do join a PhD program, they are usually paying you they pay a stipend and they pay for your school. So it's kind of like you're paid to go to school, and that's your job, is learning and participating in all of the lab activities. So that's sort of the requirements for a lot of the different types of positions. I would say pros, I got to thinking about what was good about being in a lab setting itself. What I initially was attracted to was just that you get to hear about things as people discover them. So, when you go to seminars, when you're in grad school, you are hearing about things that are not published, and they're nowhere near a textbook, because this is the current research of a lab. And one example of that, when CRISPR first came out, I remember sitting in a journal club where we were reading this article about it, and hardly anyone had heard about it at that time, but we were like fascinated that they were able to manipulate the genome. And then, now it's in textbooks, and we're how many years later? It was 2013, and now it's like 20. So 10 years it took to really get into all of these textbooks to talk about CRISPR. So I like being on the edge, like hearing about things, and going to conferences and hearing this really exciting work. The other thing I thought about is, it comes back to that question of what do you want to work on? What types of problems do you want to be involved in solving? So it's kind of indirect, but you're working, especially in basic science labs, you're working on understanding how things work. And there are probably applications or many steps down the road, but what I like is you have to be so creative, because especially with worms or with cells, you're using tools that are microscopic, or you're manipulating or setting up systems to test if this is true or not. It takes a lot of creativity, and how do you solve that problem? A big piece I enjoyed and still enjoy is how collaborative it is. It might not always feel that way, and maybe it depends on the lab too. But at least the labs I worked in, they were very collaborative. So like my colleagues were always willing to help. And even at Hopkins, I don't know that all schools have the same reputation, but other scientists and other labs would help us if we were trying to do a technique that they were very good at. I felt like they were very collaborative, and one of the students would sit down with you and share how they did it. And so I have heard that some places are very competitive and like hide their information. But that was not my experience where I was at, especially in my lab, and maybe I kind of looked out. But also then working in a field, there's also kind of the community of researchers who work on the same thing. And for SUMO, it's a pretty small field, but we would go to these joint lab meetings, and all day long, we'd be talking about SUMO. And then at the end of the day, my lab mates and I would say we had a SUMO hangover, because the entire day had been spent talking about research about SUMO. And then recently, so more in the postdoc years, then I was exposed to this other community, which is the worm community. And they're, I would say, a weird group, but all genetic model organism groups, like the fly people and the worm people, they're all kind of different, quirky. Every year they have a international worm meeting, and it's pretty wild time. They have there's some of the researchers do this comedy show. It's like 1 hour comedy show. If you look it up, you can look up worm meeting comedy show. And it's actually really funny. Like they developed this whole thing. But also they do the serious science stuff, but they also just have this kind of community of practitioners, so. And then finally, I did feel like there were times when it was very hard and there was like a lot of work where people felt pressure to stay long hours in the lab. But I would say there was flexibility it depends on the lab, but in my labs that I've been in, there was flexibility, like if you were a morning person, then you go in really early and you would do your work and then go home at like 5 or 6. Whereas some of the students that I was in lab with, they would come in late, but they would stay into the night. And actually we had jokes like we would get to work and there'd still be ice that hadn't melted yet. So we knew that Jin May had been there, like she must have been there till 2 in the morning or something. So I think you can kind of shape it a little bit. And I don't know, again, that's my experience in the labs I was in. I don't know that that's true of all. And then another positive, I think it's intellectually very fulfilling. It's very like when you go to seminars and you listen to speakers and you also listen to questions that people ask the speakers. I feel like it just raises you up to like learn or start questioning the way that you're hearing, and it just exposes you to a different way to think about things. So I always felt that it was, even though I didn't know all the stuff that everybody around me knew, I felt like I was constantly absorbing and learning from them. And especially the faculty, I felt like they were amazing in how they, again, the creative piece, like how they come up with these schemes to test some things. My boss had I felt like he had a memory, just photographic memory. He could remember what paper he read that in and what year. I mean, he just seemed to remember everything. So being exposed to that was really cool. And then the next slide is cons. And this is, I'm kind of speaking more from working in an academic lab rather than, because again, I haven't worked in industry. I think there's pressure. There is definitely pressure. Even in grad school, you feel this pressure to like, your project needs to be very exciting. You need to discover something. It has to be something that you could publish. So that feels like pressure. It's also like, sometimes you feel a pressure to get a lot of data and it kind of makes you feel like you should work more and more. In academia, I watched, especially Hopkins, they have to bring in their funding for their own salary. And so there's a lot of pressure. And when they got positions, there was this sort of time period for them where they had to get publications and grants. And I watched several people not get tenure. And for them, it was almost like shameful. And for us as students, we're like, wow, that seems pretty intense. And so that, I don't know, for me, that also kind of deterred me from wanting to be a PI, is that pressure. And then it kind of ties into this work-life balance, that being a woman in the field, that I felt like I looked around at the PIs at Hopkins and not a lot of them had kids. If they did have kids, they were kind of like older mothers, and then it just seemed like it was very difficult. And we always would ask, like when seminar speakers would come, we would ask them, when's the best time to have kids, or when do people have kids? And there was never a really good answer. People were always like, well, you just have them when you can. But that was sort of a deterring thing for me, because I didn't want to wait forever to have kids. And it seemed like there was, like you had to get to the position before you could take a break or something from that. So I think that's still a problem that plagues academia today, because if you look at who PIs are, it's mostly men in most of the R1 institutions that do research. And there's a lot of, articles that have been written about whether women are not applying for those positions or they're just not getting them. And a lot of it is that women pull themselves out of the pipeline because it does seem like a lot of pressure. So when I went through things, it was assumed that your default path was to go and try to have your own lab. But I actually just last summer had my grad school advisor came through this area, because now his son actually lives in Renton, ironically. And he was telling me now a lot of his students actually don't do postdocs anymore. So things have changed a little bit. He said a lot of them are getting positions in industry. They're wanting to make money, or they don't necessarily want the PI route. And so I think things have changed, but I went through things, you know, like, let's see, 2010 is when I graduated, so it was a while ago, so things may have sort of changed since I was there. But yeah, that's it for my slides, I think. Is that the last one? Yeah. So I don't know if you have any questions. I'm happy to answer questions. I don't know what your interests are, or either of you have any interest in lab research or anything like that.
I mean, we're both interested in medical school,
Medical
but we're always looking at like backups too.
Yeah, okay. Yeah, and I would say from, maybe we can just talk a little bit about the MBS program as, because we actually have had a couple of students who are now in medical school who did get their bachelor's. But yeah, I think the research piece helps people to apply anyway, having some experience with doing research. And there's all these opportunities to do like clinical research and things like that that can help, I think, bolster your application. So yeah, we've had a couple of students that did the bachelor's, and then one of them's at UW right now, and then one of them is at an osteopathic school in Tennessee. So yeah, I think it's good to get some research experience, and you know, some people do, MDs can do research without even needing a PhD, but that's a possibility. Which area of medicine are you interested in?
Anesthesiology,
Okay,
radiation oncology.
okay, awesome. Yeah. Well, yeah, for the recording purposes, I will just share a little bit about MBS program, because that's also one thing one of my hats is to promote that program, but I mentioned this a little bit newer. We now have had 7 cohorts of graduates, and most of them have gone to work in labs, but like I said, a couple of them have gone to medical school or school of pharmacy. So students that are interested in, you know, doing a professional school, they can look at our bachelor's program as a possibility. We have the associates, and then we have the bachelor's, which is a 4-year degree. We also now have a 1-year certificate that is a biomedical research certificate. So that is designed for people who want to get, maybe they have like, they've taken science classes for prereqs, but they want to get that lab experience. So the certificate program is 16 credits, and it's basically hands-on research. So you take these advanced methods class to learn some techniques, and then you do a capstone project where you kind of run your own project or get a chance to you know, plan experiments, carry them out, and then present your results. And so we have had a couple of people, actually the student that is one of the students is going to medical school now. She didn't get a bachelor's, but she did the certificate program before she applied. So it's a possibility to kind of add on if you're interested in getting some hands-on experience. Yeah. Any questions that you all have? You probably have a slightly different track, but
I don't think so. Yeah, get the presentation answered.
okay. Great.
Any questions from
Oh.
anybody on Zoom? Who's one? Any questions? Boss, bosses.
It's just,
It's just,
it's just
me.
yeah.
Jen.
Okay,
Jen.
well, yes,
Yes,
thanks so much for having me.
Thank you for
I
coming.
appreciate it.
thank you.
Yes.
Thank you. And this.
Stop it.